This is a short presentation on the importance of evaluating evidence, and I'm going to take you through a case study and then discuss with you how we could evaluate the evidence for and against some of the conclusions that are being drawn. We're going to have a look at the case study of Indian vultures. 3 important species of Indian vultures started to go into serious decline in numbers in the 1990s, and in fact their numbers declined so much that they fell by up to 97%, and this obviously caused a huge amount of concern because obviously there was a worry about the potential for extinction and the effect on biodiversity, but also it had a huge knock-on effect in the balance of the ecosystem and also effects upon the social structure of India. So the vultures play a very important role within the ecosystem because of the way that the cows are allowed to roam around in the countryside, and they don't get eaten. So what would happen is that the cows die, and then they just are left to decompose on wherever they've fallen, basically. And so the vultures play a very big role because what they do is they act as scavengers and they clear up the carcasses by eating all the tissues. And because there weren't so many of the vultures around, what this meant was that the cow carcasses could then become food for feral dogs. So the feral dog population has increased, and that's a huge problem for the human population because feral dogs carry rabies, and more people die from rabies bites than many other places. So the scientific community felt it was really important to try and find out what was going on and started to do some research. One of the problems they had was that the vulture population is protected and they couldn't be killed so that they could be studied. So the only way that they could do the study was to actually find some freshly dead animals so that they could investigate what their cause of death was. So in 2003 they found 28 vulture carcasses, which included a range of adults and juveniles of all the species that they were concerned about, and they tried to work out their causes of death. And from this they published a piece of research, and it outlined that there were a number of inflammatory conditions that were detected in the dead birds. Now, inflammatory conditions quite often result from having some kind of pathogen, and this is where the immune system starts to interact or react to some kind of stimulus, which is very often a pathogen. And from this they concluded that it was likely that these inflammatory conditions were caused by a pathogen which hadn't been identified, such as a virus. They did also consider the other possible causes, such as pesticides, but in fact pesticide use had declined by about 40% since the 1980s, and the tissues had been tested for the evidence of pesticides in the tissues, and they didn't identify them at all, so they certainly weren't at dangerous levels, so they felt that they could rule that out. They also considered things like lack of food, but there was no evidence that there were less carcasses for the birds to feed on than any other time. There was definitely something else causing this huge population crash. So when you're asked to evaluate the evidence, what you might be asked to do is to look at the data that you might have been given. So in this case we don't have any data. All we're really working with is the conclusions that are based upon the research. So to start with, we would look at any numbers and say how those numbers might back it up, or how they might in fact not back up the data. So we start with how the evidence supports the conclusion. Now in this instance, the main supporting evidence is the presence of several inflammatory diseases and the lack of other toxins in the tissues. So all that seems to be pointing towards the correct conclusion. Now let's have a look at how we might question this conclusion. In this instance, we don't have any quantitative data which could contradict the conclusion that's been given, but that won't necessarily make the conclusion right. Do we have sufficient evidence? Now in this instance we only had 28 vulture carcasses, which is a pretty small sample. It's not enough to be absolutely sure. They might not be representative of the main causes of death. So these might have been 28 vultures which had died for another reason, and it might then be not safe to suggest that all the other birds had died for the same sort of reasons. We don't know the time frame or the seasons that they were collected in, and that could be significant. For example, they might have all been found at a very small period of time where there was a shortage of food, or perhaps where there was a drought or other sort of conditions that are going on that might have caused them to actually die from other reasons. So we don't know that information, and that's why 28 carcasses wouldn't be considered to be sufficient, and you would certainly want to have them collected over a long period of time so that you could be sure that there weren't any seasonal variations in that data. Is the collection of the data valid? So do we have any bias in the data? Well, I would suggest that there is bias in this data at the moment, or potentially there's bias, because we don't know how those birds were selected. So it could be that they were all selected from one area, which in itself will introduce bias into that data. So did they locate the carcasses because they were on the road, for example? Or was it possible that they were located because they were all found together? So we haven't got enough information about how this data was actually selected. Have other variables been considered? So was there any sort of other thing that might be making a difference? The availability of food. Have they all had the same amount of food? Was there any hunting in the area? Is it possible that they have been hunted for some reason? Or quite a lot of the time birds of prey are not popular with farmers because they quite often are accused of hunting the young animals in, say, for example, a sheep's flock, for example. And I think that's probably a fair comment, because they probably do. Could there be another pathogen? Did they identify the pathogen there? Well, they didn't, but it could be a different pathogen. And did the time of year make any difference? So have we only got this of a short period of time, or have we managed to get animals sort of evenly distributed throughout the year to avoid the potential for bias, which is a seasonal bias? Now this leads us on to this idea of correlation does not prove causation. Now we're not really looking at correlation here because we haven't got any data, but we are trying to connect up some sort of observations and saying from those observations we think these things have happened. The absence of any other explanations does not make this one more likely, and I think that's a very, very important point to remember. So just because you can't think of any other reason, it doesn't mean to say that this is the reason. There must be direct evidence and scientific reasons for one factor to be having the effect upon the other. So normally what you would do in scientific research is that if you think that you have found a connection between two variables, you would then do scientific experiments to actually demonstrate that these are actually having an effect. So if you think that there is a pathogen, then you need to identify that pathogen, and then you need to expose the animals to that pathogen to some degree to see that they're actually being affected. Now this point can be really well illustrated using this piece of data. So we've got a graph here that shows the blood cholesterol levels ranging from 1986 through to 2010, and it shows the blood cholesterol levels of men and women over that period of time. So this is average blood cholesterol for the population. And what we can see is that the blood cholesterol levels are fairly high in 1986 through to about 1994, roughly the same. But then around about that time we see the blood cholesterol levels falling, and they fall really consistently for about 10 years until we get to about 2004, and then we can see that fall stops and in fact reverses and starts to go back up again. Now what we can't see from this evidence, of course, is we can only see the actual data, but what we can't see is any of the reasons why this might have happened. But I can tell you why this has happened, and this is to do with the birth of Justin Bieber and the invention of Facebook. And you can see quite clearly on this graph that the benefits of Justin Bieber's birth has been cancelled out by the appearance of Facebook. And I can show you here what I mean by Justin Bieber being born in 1994, where the cholesterol levels are starting to fall, and just at the point where it starts to go back up, Facebook has been invented. So it's not hard to see that actually these two events have triggered the changes in blood cholesterol in the general population. And hopefully this is a nice illustration of how you can take unconnected events and actually link them in a totally unscientific way to another more significant event. So what I want you to think about here is it's not enough just to sort of link up two things and say, oh, that happened and then this other thing happened. They are just coincidence. For example, you quite often get increases in two factors, but it's more to do with perhaps a general increase in the population rather than there actually being a real increase in the actual prevalence of that condition. And so in this instance here, blood cholesterol levels have fallen, but very unlikely to do with Justin Bieber. So to summarise, a good evaluation of a conclusion needs to include reasons for and against the conclusion, and if possible, use data and statistics. So if you've done a statistical test and it shows that there is a significant difference or that there isn't a significant difference, then use that data and comment on it. It could be, though, that that's not the whole story and that actually other bits of data are suggesting an alternative. A very good example here would be where you have standard deviations which are very big, which indicate that the means of the data aren't particularly representative of the data as a whole. Consider other confounding variables, anything else that might have had an impact upon this data that might actually be affecting the way that the data is being collected. Evaluate the validity, repeatability and reproducibility of the data. So by validity, what we mean is, have all the control variables been controlled? If you're investigating something in an environment, have you measured all the other environmental factors, the abiotic factors, for example, like temperature and pH of the soil? If you're doing a lab investigation, have other factors, for example, light levels, temperature, concentrations, have they all been taken into account? Is the data repeatable? Does that mean, and that means have they done more than one repeat of each investigation, and does the data look very similar, and which would make it repeatable? Could somebody else get that same data, making it reproducible? You need to consider alternative explanations. Sometimes the obvious explanation isn't the correct explanation, and maybe there is in fact another reason for this data, and we need to consider what that might be. So think about what the other control variables are and ask yourself, could any of them really be the main factor involved in this investigation? Do settle on one over the other, but don't include any certainty. So if you decide that you favour one conclusion over another, then it's okay to say that. But please try to include a level of uncertainty in your answer, because you're showing that you can take the data into account. And therefore that would perhaps lead us on to recommend further areas for research which might remove some of the uncertainty from your mind. So how did this mystery conclude? Well, in 2004, a team of American researchers identified the presence of a drug called diclofenac, which is an anti-inflammatory, it's a painkiller, and it has been used to treat domestic animals as well as humans, in fact, since the early 1990s. And this drug is lethal to vultures. Lots of other animals are treated in a positive way and actually, benefit from the pain-killing effects and the anti-inflammatory effects, but in the vultures it was really problematic and it was actually killing them. And they were able to demonstrate that this drug was having a lot of damaging effect upon the organs. So since then, diclofenac is now banned for veterinary use in India, Pakistan, Nepal and Bangladesh, and this has actually reduced the decline of the vultures, although recovery of this is going to take a lot longer than. So we can just see there a little bit of data on the numbers, and you can see that the population has been going down and down until about 2008. So and that's because, of course, diclofenac would have been still in the population of cows and animals for quite a long time, so that the vultures would still have been vulnerable to its exposure to that drug even after it had been banned. So just now we're starting to see the numbers starting to go back up again, and obviously that's where the presence of the drug within the tissues of the newly deceased cows have actually got far less diclofenac in them than they would have had before. So thank you very much for listening to this. I hope you find it useful, and it will give you some guidance on answering questions on evaluating evidence.